Hi, my name is Anupon Lahavisit, and I am a junior principal investigator in Nagoya University in the Institute of Transformative Biomolecules. So my research is looking at how plants they produce and how they sense quinone. So quinone are special chemical molecules that are produced by all living organisms, from microbes to animals to plants. In animals, the molecules are used for many purposes, and the quinone molecules can be quite simple or very complex in nature depending on the nature of the molecules. In plant, we know that quinone are produced by many plant species. So for example, when an apple turns brown, that is because the chemical catechol is converted to quinones, which cause the apples to go brown. Perhaps the most well-known example of how quinone is used as a signal in plant is from parasitic plants. Parasitic plants are plants that parasitize other plants and therefore stealing nutrients from the host plant. To do that, the parasitic plant needs to germinate, and once it germinates, it would need to attack and produce a specialized feeding structure, which we call the hostorium. For plant to form hostorium, it needs a signal, and this signal is actually come from a host plant, and this signal is actually a quinone compound that the host plant is produced. Once the parasitic plant recognizes these molecules, it will begin differentiating its root into a specialized feeding structures. In my previous research, I was able to identify how plants sense these signaling molecules and what pathway it involves. Interestingly, all plant species utilize similar receptor, which I named CARD1 for cannot respond to DNPQ. However, even though it goes through the same common signaling pathway, the resulting outcome is different. For plastic plant, the signal is used to cause development of hostorium, which is required to attack other plant species. However, for non-plastic plant, this signal is actually used for plant immunity. Here in ITBM, my research will be to find out whether plants produce other quinone compounds and whether these are also signaling molecules in plants. As well as that, I want to identify if there's any other quinone signaling pathway in plant. Eventually, I would like to manipulate quinone signaling in plant. And if we can manipulate the signaling response in plant, perhaps we can choose the response that we desire. For example, we can stop plastic plant from making hostorium to parasitize other plant, or we can enhance normal plant immunity response, for instance.